A groundbreaking three-dimensional cell center model has been developed. This new model overcomes the limitations of previous cell center models in 3D space. Detailed information on this model is available in the references. The source code is accessible for download on GitHub. Living organisms develop macroscopic forms through the interactions of numerous cells at the microscopic level. Mathematical models and simulations are crucial for understanding how these microscopic behaviors give rise to complex macroscopic forms. There is a need for a new mathematical model capable of handling a large number of cells at an organ scale. Simulation challenges arise when the number of elements is extremely large, as computational load increases, making analysis impractical. The 3D vertex model represents cell shapes as polyhedra and allows mechanical analysis at the cellular level, but is computationally intensive for large-scale organ-level analysis. The cell center model is computationally lighter, but has limitations in recovering cell shapes and cannot be applied to curved tissues in 3D space, making it suitable only for 2D analysis. In the cell center model, cell shape is determined by Delaunay triangles connecting cell centers and their duality with Voronoi diagrams. On a flat surface, Delaunay triangles and Voronoi diagrams have equivalent information. Issues arise when constructing Delaunay triangles on curved surfaces. Constructing a Voronoi diagram from a Delaunay triangle and reverting back doesn't necessarily return the initial Delaunay triangle. Ideally, this should be an identity map, but inconsistencies occur. Additional complications emerge with obtuse triangles. Calculating Voronoi diagrams while maintaining adjacency becomes difficult. Circumcenter points of obtuse triangles are outside the triangle. Ignoring adjacency enables Voronoi segmentation, but results in adjacency changes due to mathematical algorithms, which is not suitable for simulating morphogenesis. To bypass the complexity associated with Delaunay triangles and Voronoi diagrams on curved surfaces, the center of mass is utilized to represent cell shape. Unlike the circumcenter, the center of mass is always inside the triangle. With three cells in contact, a triangular element can be constructed by connecting neighboring cell centers. The epithelial sheet is represented as a network of triangular elements formed by connecting adjacent points. This framework allows for the mathematical representation of cell shape, motion, intercalation, division, and apical constriction. The inertia term is omitted to simplify the equation of motion for cell I. The resulting equation incorporates an energy function E, and energy E is the sum of mechanical energy from the network of triangular elements and constraint energy. The constraint energy ensures satisfaction of the constraints regarding the cell center. Epithelial morphogenesis involves cells changing their positions actively or passively. Cell rearrangements are modeled through changes in adhesion between neighboring cells. A simulation technique called flipping was used to model these rearrangements. Flipping involves replacing the edge between two cells with an edge connecting two different cells. The flipping is performed only if the distance and expected distance after the flip meet certain conditions. The positions of the cells do not change, only the connections between them. A cooling time is introduced to prevent continuous flipping between the same connections by fixing the newly formed connection for a certain period. The study investigated the effect of cell rearrangement via flipping in simulations. Simulations were conducted under two conditions: infrequent flipping and frequent flipping. The cell sheet was subjected to compression in one direction and then allowed to relax. The focus was on differences in time variation of two-directional strain within the plane and the final shape of the cell sheet. Through this approach, the model could represent both elastic and plastic deformation based on the frequency of cell rearrangement. The model developed in the study does not include a parameter for sheet thickness. Instead, it utilizes a dihedral energy term to account for sheet stiffness effects. The quantitative validity of the dihedral energy term was evaluated through simulations of sheet buckling caused by cell proliferations. The simulations were performed under conditions similar to those in previous 3D vertex models for comparison. The folding pattern observed aligns with those from previous studies using 3D vertex models. The wave number's dependence on the out-of-plane deformation constraint constant follows a power law. The power exponent of this relationship approaches the theoretical value of 0.25 in the continuum, which is consistent with mechanics and buckling theory. Simulations were performed to investigate the mathematical expressions of apical constriction in the model. The model incorporates spatial patterns of apical constriction from a previous study. A key evaluation criterion is whether invagination forms at the center of a hexagon. A previous study using a 3D vertex model showed different concave shapes depending on the case, A or B. The current model successfully reproduced the same results as the 3D vertex model. Though the model does not depict the 3D shape of cells, it demonstrates the tissue scale deformation effect equivalent to changes in cell shape due to apical constriction. This is attributed to the design of the apical constriction energy on the neutral plane of the epithelial sheet. The developed model is highly efficient, enabling quick computations. It allows for simulations from cell proliferation-induced buckling to the emergence of new folding deformations resembling folding a handkerchief. However, the current model has limitations and cannot handle multi-layer structures. In such cases, 3D vertex models might be more suitable. The model excels in working with monolayer cell sheets. The model incorporates the cell center point, allowing the integration of self-propelled particle models for considering collective cell migrations. We encourage the use of this model for further research and exploration. For collaboration and joint research, please contact Professor Inoue at Kyoto University.